Now that we have finished our discussion of lipids, we now move into Chapter 11, where we look at how lipids are incorporated to produce our membranes. The first thing we need to consider is, why do we need a membrane? A membrane is going to allow us to separate the internal from the external environment. Not only do we separate with a membrane on the very outside of our eukaryotic cells, we also have internal organelles that have their own membranes to further separate different areas. Membranes are composed mainly of lipids or lipid-based structures, but the membrane is not just made of lipids. It is composed of both lipids and proteins. A nice way to think about it is that the lipids provide the structure for the membrane, while the proteins actually do the work within the membrane. The proteins are responsible for receiving signals, moving things through the membrane, and other things that we'll discuss as we move through the chapter. Membranes themselves, the lipid and the protein components, as I said, define the boundaries of our cell, but they also work to allow selective import and export, retain specific things within the cell, allow external signals to be transmitted into the cell. When we move into the next chapter, we will discuss the transduction of the signal, how it gets from the outside of the membrane into the intracellular space. Within the cell, the membrane allows us to compartmentalize. The membrane allows us to transmit specific nerve signals, produce a proton gradient within the electron transport chain of the mitochondria so we can produce energy, as well as many other things that are not provided on this slide. When we start thinking about the features of the membrane and how a membrane looks, we need to recall our understanding of the hydrophobic effect. Due to the hydrophobic effect, the lipid molecules are going to spontaneously aggregate together. Once they aggregate together, they're going to form this lipid bilayer. Now, the membrane itself is not just going to be a perfect, even structure of lipids. It's going to have, as I said, some proteins, but you may have one lipid that's preferably found on the inside of the cell, where another is found on the outside of the cell. Your lipids that are attached with carbohydrates are always on the outside of the cell. So there's some real common features that we can state about all of them. So as we remember our discussions of hydrophobicity from our earlier chapters, we remember that if a single lipid molecule, single hydrophobic molecule, is left in an aqueous environment, we will get a very ordered pattern of water molecules around that lipid. That is not a favorable reaction. It decreases the entropy of the environment. So what lipids do, the hydrophobic molecules will allow their hydrophobic regions to interact with each other, decreasing the amount of the water cage and therefore increasing the entropy. So if we had a simple fatty acid where we had a more hydrophilic head and a hydrophobic tail, that lipid's going to have this more cone shape to it. If put into an aqueous environment, they're going to spontaneously aggregate into this micelle structure. This is not what we see in a membrane. The membrane bilayer is composed of lipids, mainly phospholipids, but some others that we'll review here in just a moment, that have a more cylindrical shape. That's going to allow them to form a bilayer, and if the bilayer continues to form, it will ultimately form a vesicle, and this is how we end up getting our membrane around cells. So now we move back to thinking in a little bit more detail about the composition of the membrane. We cannot just state something definitive, that a membrane is made of one type of phospholipid with a certain percent of protein and a certain percent of cholesterol. It doesn't work that way. The location of the membrane, the tissue that the membrane is in, is going to help dictate some of that composition. So just to show you some differences that we can see, if we look at down here at this table, the myelin sheath you see is got a pretty good concentration of phospholipid and cholesterol compared to protein. if we compare that to some of the other cells, you see the other cells have a much higher quantity of proteins. And if we think about the function of the myelin sheath is the wrapping of the cell around the nerve cell. It mainly serves as a protection function, an insulation, so we don't need as many proteins to do any work. Where if we compare that to a liver cell or some of these single-celled organisms, their membrane has to do a lot of work, so they need a lot of proteins. Looking at this a little bit closer, we can look at some of the cholesterol comparison within different membranes in different locations within human cells. If you look at the plasma membrane, it has a pretty high concentration of cholesterol. You compare that to something like the rough endoplasmic reticulum, the ER is going to have a much lower concentration of cholesterol. So if we think about why would that occur? The function of the rough endoplasmic reticulum is to allow proteins to be produced and then little vesicles to pinch off, moving that protein next to the Golgi and so on and so forth. Those membranes are less static. They're constantly being made and used and pieces broken off, so we don't want quite as much cholesterol. The cholesterol makes the membrane much more rigid. Plasma membrane, we need a little bit more rigid. It's protecting the entire integrity of the cell. And we can make some similar comparisons with some other lipids as well. The membrane bilayers, if we compare the outside to the inside monolayer of the lipids of the membrane, we can see differences as well. When you look at the sphingomyelin, very high concentration in the outside. You also see that with phosphatidylcholine, which is a specific type of phospholipid. Those are more commonly on the outside monolayer. When we look at phosphatidylethanolamine, which only differs from phosphatidylcholine in the X group, the extra group on the phospholipid, we see very little of the ethanolamine in the outer layer, but a ton on the inner layer. So you can actually figure out which type, whether you're looking at the inside or the outside of the membrane, strictly by the type of lipid that you're finding in highest concentration. There's a very specific reason that this occurs. We put all of our positively charged molecules on the outside, giving the outside a much more positive charge. The phosphatidylserine has to be on the outside of your platelets to activate blood clotting. Well, if you think about the process of blood clotting, it requires cells to interact with each other in a specific way. Well, we don't want all of our cells to do that when it's not specifically called for. So most cells have phosphatidylserine on the inside, where our platelets have a lot on the outside. Phosphatidylcholine helps to mark a cell for destruction. So in specific cells, phosphatidylcholine is not moved to the outer layer until apoptosis has been signaled. So a lot of communication with the cell and other cells can occur simply by the type of lipid that is being displayed. If we look at this picture, it gives us a nice graphic demonstrating the differences in the type of lipid present throughout the cell. If you notice, the phosphatidylcholine is in high concentration within the Golgi, right? But as we move further to the outside of the cell, we no longer see the phosphatidylcholine. The phosphatidylcholine is replaced more with phosphatidylethanolamine, phosphatidylserine, as well as some sphingolipids. If you recall, I told you in the last slide, the phosphatidylcholine signals apoptosis. The phosphatidylcholine is mainly within the cell. Now, it's in the outer leaflet within the cell, but it's not in the plasma membrane. Now, making sure we don't spend too much time just looking at what's going on with the lipids, let's look at a more realistic picture of what's going on with a membrane. We have adopted the fluid mosaic model for the structure of a membrane. Fluid because the membrane is not static. The molecules are moving around within the membrane. And mosaic because there's lots of different things within the membrane. Lipids, proteins, sugars, several different things. Now let's take our attention a little bit more to the proteins found within the membrane. Proteins serve a lot of functions. Three of their most important functions are to act as receptors to detect signals from the outside. They can act as channels to allow specific transport of molecules through the membrane, and some act as enzymes themselves. One that we'll spend a lot of time covering in our next trimester is the ATP synthase, which allows us to produce ATP. The proteins within the membrane exist in one of three ways. We can have an integral membrane protein like this one that spans the entire membrane. Typically what's on the extracellular surface looks very different than what's on the intracellular surface, but it still spans the entire membrane. We have the hydrophobic interactions in the middle and the more hydrophilic interactions on the edges. To get an integral membrane protein out and to break those away from the membrane, you have to have some sort of detergent to interrupt those hydrophobic reactions occurring within the middle with the more hydrophobic amino acids and the lipids. A peripheral membrane protein is going to interact either with the hydrophilic portion of one of the integral membrane proteins or simply interact with the more hydrophilic head groups of the lipids. To release a peripheral membrane protein, you don't need a detergent to disrupt the hydrophobic interactions. Then you'll only need to change your ionic interactions. So pH or salt can change the charges enough to get those peripheral membrane proteins to let go. Amphitropic membrane proteins interact electrostatically, or they can be lipid anchored. Most of these proteins are going to really have a lot to do with regulation, and we'll look at a few of these in the next chapter. This slide shows you a much more realistic look at what the protein looks like within the membrane. If you recall from our early discussions, we take the different amino acids and we allow them to form this specific secondary structure, alpha helix or a beta sheet. So what happens within the membrane is the hydrophobic residues will lay just correct so that they are on the outside of the alpha helix to interact with the hydrophobic part of the membrane. Around the edges of the alpha helix, near the outside of the membrane, that's where the more hydrophilic amino acids are going to be located, charged amino acids. Looking at the primary sequence of the protein, you can see as we move into the hydrophobic region, we've got a lot of hydrophobic residues. In the outside of the membrane, we have more polar, hydrophilic, and even charged residues. The integral membrane proteins exist in a couple different types. Type 1 and 2, shown here, are single-pass proteins. They differ by their direction. Type 3 is one protein that passes through the membrane multiple times. We see this a lot with our channels. Type 4 and 6 are lipid-anchored proteins. And then type 5 is you get multiple passes through the membrane, but multiple proteins allow this to happen. This graphic shows you a little bit more. It's got the space filling model for the proteins, and it allows you to see all of the white residues. Those are hydrophobic residues. So you can see they're highly concentrated within the hydrophobic region of the membrane. The more charged residues are in blue, and you can see the majority are outside the membrane. Another thing you can see from this picture is that we have a lot of tryptophan and tyrosine residues right at the boundary of where the nonpolar and polar molecules meet. If you think about the structures of tyrosine and tryptophan, they are amino acids that have a nonpolar region and a polar region. So they're arranged in a perfect environment for their structure. Most of the integral membrane proteins that we will interact with as we move into the next chapter are going to be membrane proteins that have alpha helices. But we can have beta sheets that allow us to span the entire membrane. The beta sheets are much more extended than the alpha helix, so it takes a lot less amino acids. You're looking at about 20 amino acids to get an alpha helix through the membrane, where with the beta sheet it's only 6 or 7 residues. One topic that we need to discuss while we're discussing membranes that is often overlooked in undergraduate biochemistry are lipid anchors. There are a lot of membrane proteins that exist covalently linked to the lipid molecule. Some of these are there for structural purposes, others are there for a specific function. So I'm going to show you some of the lipid-linked proteins and some of the functions of those. We see a lot of lipid-linked proteins on the outside of the cell that have covalently attached sugars. These are GPI anchored proteins. And then on the inside surface, we see other proteins that are lipid linked, and you notice they're all linked to specific amino acids, and we give very specific names to each of these. So what could this possibly be for? This allows, well, I thought it was the next slide. I'm going to show you what those lipid-linked proteins are for in just a moment. Okay. So let's look at the actual physical properties of the membrane. I told you it's called the fluid mosaic model because it is a fluid membrane. Things can move. There are some changes that can occur within the phases of the membrane. At lower temperatures, the membrane can exist in a more gel-like structure where the molecules are moving a little bit less, so it's a firmer membrane. But as you get to higher temperatures, the molecules are a little bit more mobile and you get a more lipid structure to your membrane. There are other things that can affect the phase of the membrane. The more saturated lipids you have, you're going to more likely have this gel-like state because of the nice packing of those lipids. When you have more unsaturated fatty acids, remember with a unsaturated fatty acid, you get that cis double bond, so you get kinks. That's going to make them not pack quite as tightly, so you can get a more liquid membrane. So if you think about it, if you take a membrane that has this more saturated fatty acid look to it, and you start raising the temperature or lowering the temperature, you can actually adjust whether it's more gel or more liquid by changing the type of lipids that you have. The body is constantly making and breaking down lipids, so it's a constant changeover with the lipids within the membranes of the cell. Okay. So just a few notes for you. Some of this is kind of what I've already said to you. So anytime you have shorter and more unsaturated fatty acids, you get a more fluid membrane. So at high temperatures, that's when the cell will make accommodations by putting in saturated fatty acids. Because at the high temperature, it's more liquid, so you put more saturated ones in there, it packs tighter, so it's not quite as liquidy. If you go low temperatures, the membrane might get too gel-like. So then you need to start sticking some unsaturated lipids in there so it doesn't get too firm. It can maintain some of its fluidity. And I found this picture online. I thought it would really help you see. Cholesterol itself, if we just think about cholesterol, when you stick it in a membrane that has a bunch of saturated fatty acids, it gives you a more rigid structure. And it also helps with the unsaturated fatty acids because that cholesterol fits perfectly into the kink that's formed by the unsaturated fatty acids. So anytime you have sterols within the membrane, that's going to make the membrane much more rigid, much more gel-like. The lipids can move laterally, uncatalyzed very quickly within the membrane. Proteins can also move this way. But when we start thinking about the move from one side of the membrane to the other, that becomes much harder to accomplish. We can, however, see an uncatalyzed transfer from one side of the membrane to the other by the lipids. This is, however, an extremely slow process and does not happen as often. There are some enzymes, and remember enzymes are proteins, so there are some proteins within the membrane that can actually catalyze the transfer, the translocation of a lipid from one side to the other. They have very cute names. Flipases are enzymes that allow the flip to occur. This requires energy. Flopases go in the other direction from inside to out. They use energy. Now, scramblases allow you to move lipids, but that one doesn't require energy. That moves with the concentration gradient, depending on the type of lipids that you have. So now we're back to what I was discussing earlier with what occurs with some of these lipid-linked proteins. So there's areas within your membrane called membrane rafts. Membrane rafts occur where you have high concentrations of glycosphingolipids. Glycosphingolipids always have a much longer than usual fatty acid tail. So when you get a region of the membrane like this, where you've got the longer fatty acid tail, you can see it's going to cause a thick spot in the membrane. Those thick areas are where you're going to see a lot of your lipid-anchored proteins. Caveolin is a very common one that allows the membrane to bend or form a curve. I think I have a picture of that, yeah. So if you have quite a few of these areas where you have this Caveolin interaction, that protein is going to cause curvature. The curvature is important in allowing the membrane to begin to form a vesicle so that you can get it to pinch off and leave. We see this with exocytosis and things like that. That's not the only protein that can do that. We have a lot of proteins that can interact in a specific way with the membrane, causing the membrane to want to form a curvature. That curvature leads to the ability of a membrane to have membrane fusion. And as you can see from this slide, we have quite a few examples of that occur within the human body on a regular basis. The vesicles formed from Golgi must leave by exocytosis. Other things come in by endocytosis. Unfortunately, viruses can enter our cells. But fortunately, humans can reproduce because the sperm can enter into the egg. So this membrane fusion is pretty important. And it all goes back to these specific areas that are marked as places that's easy to occur because you have very specific proteins in these thicker regions, these membrane rafts. So now we have a pretty good idea of what the membrane looks like. The membrane is fluid. We can adjust the fluidity of the membrane by the type of membranes, the temperature, the presence or absence of cholesterol, quite a few things about the structure of the membrane. Now let's transfer our train of thought to transport. How can we get things across this membrane? Well, some things are pretty easy to get across the membrane. If they're very small and hydrophobic, they can simply diffuse through the membrane themselves. They can go through the hydrophobic middle all by themselves. But any molecule that is not small and hydrophobic would really be fighting to get through that hydrophobic region of the membrane. We have to have protein transporters to make this happen. So there's different types of transport. I'm sure you've learned in your classes in the past, but let's review them for the terminology. Simple diffusion is when a molecule that is nonpolar, small, moves down its concentration gradient through the membrane directly. Facilitated diffusion still allows a molecule to move down its concentration gradient, but we have to have a protein because this molecule is not nonpolar. It's a polar molecule. Active transport allows a molecule to move against its concentration gradient. Primary active transport requires energy and just pulls the molecule against its concentration gradient. Secondary active transport links the move against the concentration gradient with another move that's going with the concentration gradient. And then ion channels just form specific holes allowing the molecule to move. So a little bit of biochemistry around this. If we consider just a polar molecule, it's going to be surrounded by water molecules. In order for this to just magically move through our hydrophobic membrane, all of these water molecules would have to disassociate. The polar molecule would have to go through the hydrophobic region, then come back out and be rehydrated. These reactions would have a huge free energy to overcome. So what we do is we allow, when this polar molecule has to let go of its water, it can interact with polar residues of the transporter integral membrane protein. This lowers the delta G, allowing us to move molecules across the membrane. Molecules can move singly, one at a time, in one direction. That's uniport. More than one molecule can move together in the same direction. That would be a symport. Or more than one molecule can move in the opposite direction, and that would be an antiport. Both symport and antiport are co-transport systems. So let's look at a few examples just to make sure we have a good understanding of what is occurring as we transport molecules across the membrane. This particular figure shows you an example of a glucose transporter. So what we see when we look at the whole picture is we have a molecule that has multiple alpha helices within the membrane and then charged residues on the outside. When we look at it from this angle, doesn't quite give away the scenario. But if we look down one of the alpha helices, what you can see is that the alpha helix forms so that the nonpolar amino acids are on the outside of the helix interacting with the nonpolar hydrophobic fatty acid tails, and it forms a little pore. That pore is going to be lined with polar residues. So the glucose that this transporter is designed for has the appropriate size pore with polar residues for the glucose to interact. All of this is protected on the inside of an alpha helix that is surrounded by hydrophobic residues to interact with the hydrophobic fatty acids of the membrane. Let's look at the same glucose transporter and think about it as it's really more than just that pore. What happens is you have a protein that's in a certain orientation. This strange looking structure here is actually the alpha helix. When the glucose binds those hydrophilic residues within the inside, that causes a conformational change in the protein. That moves the glucose now to where it can move into the cell. When the glucose lets go, we get a conformational change back to the original structure. The entire time this cycle is occurring, hydrophobic residues are still on the outside and hydrophilic residues are on the inside. So that was a uniport. Let's look at an example of an antiport. This is a reaction that we should all be familiar with as we've worked with it already this semester. So we understand that carbon dioxide is not very soluble in our blood, so we convert the carbon dioxide to a bicarbonate molecule. Now we talked about one reason we do this is to affect the pH, and that allows us to change the conformation of hemoglobin so that we can bind and release the oxygen. But there are some other things occurring. In order for this reaction to occur, the carbon dioxide has to come into the red blood cell, where the carbonic anhydrase enzyme converts that into bicarbonate and a proton. Well, that bicarbonate now needs to come back into the blood itself, where it can be dissolved, help buffer, and so on. But we can't just keep sending negatively charged molecules out into the blood, or it would offset our charges. So every time a bicarbonate leaves, a chloride ion comes in. So we get an exchange of our negative charges. So here's some active transport examples. So just kind of cartoony drawing first, and then we're going to move to a real example. So here's the primary active transport, where we're moving this one molecule against the concentration gradient. We use energy. But something else we can do is we can move this molecule out. Okay, same scenario as the first. That is active transport. But then we can allow this molecule, this red molecule, to flow back in. Flowing back in will give us the energy needed to get the second molecule to flow in. So primary active transport we can look at the sodium-potassium ATPase. This is a P-type ATPase because it uses energy. The sodium-potassium pump, or ATPase, is an incredibly important protein found within your membrane. This protein establishes the resting membrane potential of the cell. It keeps the inside of the cell more negatively charged. Every time this protein has a cycle, 3 sodium molecules are moved out while 2 potassium molecules are moved in. So even though we're not moving any negative charges, we're taking 3 positives out and only bringing 2 positives in. This changes the charge state on each side of the membrane. What is actually occurring, even though I don't have a nice graphic for you to see, is this protein has a binding site for the sodiums on the inside of the cell. When the sodium binds, causes a conformational change, moving the sodium out. In the new conformation, you now have a binding site for 2 potassiums, and so on and so forth. It also has that similar structure to the glucose transporter where you have alpha helices that have hydrophobic residues to interact with the hydrophobic fatty acids, and more polar residues on the inside, and even a charged aspartate to allow the sodiums and potassium charged molecules to interact on the inside. Some cells use the movement of the sodium and potassium to help other things occur. So we can take, so this picture here shows you, this is the inside of the intestine. So you've just eaten and you've got some sugar in your intestines that you need to get into your blood. Well, we only want this to move in that direction. We don't want sugar from the blood going back into the intestines. So what we do is we have a co-transport, a facilitated diffusion of the glucose and sodium into the inside of the epithelial cell. That sodium is then pumped out by the sodium-potassium ATPase. So since you're always moving this sodium out of the cell into the blood, that allows this facilitated diffusion to continue pulling sodium into the cell. That continues to then pull glucose in and move glucose into the blood. Leak channels are sometimes a little bit harder for people to really wrap their head around. The way the potassium leak channel works, and I'm sure you've talked about these when you went over muscle contractions and things of that nature. This particular protein has 4 different regions, all alpha helices, that sit in the membrane to create this perfect little pore. That little pore has specific residues that interact just so the potassium surrounded by water molecules fits perfectly into this pore. The sodium itself doesn't fit into the pore correctly. So even though this really is sort of just an open channel, only potassium has the correct structure that it can simply flow through. As we move into our next trimester, we'll spend quite a bit of time going over how ATP is produced, but just to introduce this to you now, those we were just looking at were P-type. This is an F-type ATPase. They are very complex. They are used to pump protons across a membrane, creating a proton gradient. And in the mitochondrial matrix, this is how we produce energy. So we'll go over these a lot in the next few weeks. And then we finish up today showing you this specific type of transporter called an ABC transporter. ABC transporters are used to drive very specific substrates across membranes. We have these that move all sorts of things across membranes. For the most part, they are very important to us. They move molecules in the direction we want them to move. But believe it or not, they actually have been bad for us. As we have developed drugs, and specifically several cancer treatment drugs, we have figured out the reason the cancer treatment was not successful in humans, where it may have been in some of the trials, where these ABC transporters on the outside of our cells were recognizing these drugs, these molecules, thinking they shouldn't be in the cell, and then they were spitting them back out of the cell. So just another side of what the proteins can do. So overall, spend some time looking at what makes up the protein, what makes up the membrane, like proteins, lipids, what are some of its properties, and then how does transport across the membrane really work.